Okay, so microphone is on now. We'll start. So we were looking at brain waves. Mainly gamma waves, because it's related to all these cool things. Without explaining how to plot this, I told you about power spectral density and time frequency plots. We'll spend a lot of time actually figuring out how to plot these and later in the course. So in the first class, I was just telling you about gamma oscillations and some monkey recordings where a monkey looks at a big stimulus whose orientation and spatial frequency is what we vary. And then I shared the data um, and the codes to which allows you to visualize this. That was the first and the second part. Then we started talking about the EEG data set. Yeah, so this is what we did in the first two classes. This is still monkey stuff, the traveling waves and cross frequency coupling. In the last class, we started with the human EEG data set, and I told you about this first data set, which is already publicly available on aging and mental disorders, where, yeah, so the data and codes are all here, where essentially people from different age groups viewed gratings that generate gamma oscillations. And what we found is that as you age, these gamma waves become weak. As people who have MCI or early AD, their gamma is weaker than corresponding age and gender matched controls. It's very consistent for people who, it's very different across people, but very consistent for a given person. And these are other things that we have done, which you guys can look at to get ideas about what you can do for the meditation data set also. Then we talked about the actual meditation data set on which you guys will work. So the codes are all here, and we looked at some preliminary results. Not preliminary, I mean, it's a proper result where we saw that when the eyes are open, meditators have more power than controls, even when they are not meditating. And this is occipital and frontotemporal groups. And when they meditate, the power shifts up further. Then, and in G2 protocol, which is where they're viewing at gratings after meditation, meditators have more gamma than controls. That was our key finding. And this actually happens even before they meditate. So it's actually a sort of a trait change because of this continuous long duration spanning years where they have meditated and supposedly it has changed their brain. Okay. And then we looked at this M2 protocol where they are meditating while looking at gratings. And there, during epochs when there is no stimulus on the screen, meditators have this frontal broadband gamma, stronger than controls. When they are looking at gratings, they have more occipital gamma than controls. And this is where we stopped in the last class. So today I'm going to actually show you how to use the coding, the codes and the data to which will allow you to actually play with the data yourself. So this is how we have to do things. So I shared this folder called segmented data with you. You guys should have all got the link, and you have to keep it within a folder called data. So this is how it should look like. So you have a folder called BK1. This is Brahma Kumari's one. Within that, there is a data folder. And within that, there is segmented data that's shared with you. We also have, I also have raw data, but you really don't need this. Segmented data is all you need. After that, you need to download or preferably clone certain folders from repositories from GitHub that are useful for running this project. Unless you have these, the codes will not run. Common programs we already have seen in that previous monkey resources folder. It's also there on GitHub. So you can clone it and keep it. I haven't done it in this because it's there in that monkey resources folder. As long as it's there in MATLAB's path, it doesn't matter where it is kept. Similarly, you need to have this folder called montages. This I have kept here. And when you download or clone these programs, you will have a programs folder here. It can have a different name. I think if you clone it by default, it will just have this name, Project Dhyan BK1 programs. It doesn't matter. As long as, these the paths are there for MATLAB to find, it's all good. So we also need certain software. Chronux is what we have already downloaded. For that monkey resources part, you can get it from this here. And EEG lab is required, and a field trip for some connectivity analysis that we will get to later. So it's a good idea to keep both. Set up my alarm 9:55. So once you do this, just get the slideshow. Then you will have a folder like this. And within this, you will have data here, segmented data. Raw data is not needed. If you look at segmented data, these are the different subjects. So let's see how many we have. 78 items. So these are the 78 good subjects that we start out with. Within, for each subject, you will have these details here. These are the 8 protocols. I closed 1, I closed 2, I closed 1, EO1, then G1, then M1, and all that. And within this folder, again, you have segmented data, and this is the raw data that you'll be needing. Okay, so let's see if I open this, what I get here. Okay. I had to do this way. It's going to try to load this. See if it loads it or runs a new. Okay. So notice here that it's an array of size 120 cross 2500. 120 is the number of segments. Okay, so here there is in E01 where we have this, the subject is just looking at a screen for 5 minutes with eyes open. There's nothing else. So what we have done is simply this 5-minute segment we have divided into segments of 2.5 seconds. Okay, so this is 500. So this is 300 seconds. And we simply divided into segments of 2.5 seconds. Its sampling rate is 1000. So 2.5 seconds is 2500 samples. So you will have 300 divided by 2.5, which is 120 such segments. This is done because later when we are going to show them gratings, it has a similar architecture where you show a greeting for 1.25 seconds and then there is a blank for 1.25 seconds. And this just goes on for 5 minutes. This is what you have in the G1 protocol. This is EO1. So again, now we will we will now segment it based on the onset of the stimulus. So 1.25 seconds this way and 1.25 seconds after that. So we have preserved the same logic even for segments where the subject has eyes open or closed. They all have the same duration here, 2.5 seconds. Okay. So that's all segmented data. Now, let me just tell you a little bit about this programs folder. Okay, so to run this code, you just need to have certain things in MATLAB's path. So here you will go environment set path. And let me remove these. These are not needed. Something else I was doing yesterday. Simply means that you add with subfolders, this EEG lab subfolder, it will add all these. Similarly, you have the montages and the programs. Okay, once you have this, you are all set. In addition, common programs are needed and Cronux are needed. This we did when we were playing with the monkey data. Okay, once you have all of this, you are all set. So the way this sort of coding architecture work is as follows, that you have some common analysis codes. These are codes that are required for all the other projects. So you should have this in your MATLAB add path as well, but the other ones are not needed. And every different project like analysis of power, analysis of connectivity, analysis of sources, and it's essentially a different folder. Okay, so when you do your project in your grant proposal, I'll expect you to make another folder within this. Okay, so to do that, obviously, you can't play around with my repository. So what you should do is what is called fork, to create a fork of this repository. So this is now me on GitHub, where all these codes are. Project. And you will, there's a fork, there, it's a fork request. So essentially what this does is that it creates a copy of the same thing under your username. Okay, so what is happening here is that, so this is me, this is Supratim here. This is on GitHub. So this is my GK1 folder or project Dhyan, whatever it's written. And I have cloned it to a local computer here. Okay, so this is a local computer. In this case, on my desktop, I have a copy here, programs. If I make any modifications, I can push it up. If so basically, I have very tight control of what changes I have made at what time. So this is how you version control something. What I'm asking you guys to do is to actually fork this repository. So this is some other username, Rahul. So Rahul will have his own repository on GitHub. So he will have a copy of this. This is what he should be cloning onto his computer. Then you can do whatever you want. If you make a mistake, it's all right. You can reclone. It doesn't matter. Now, once then I expect him so to add whatever he wants to do, whatever new analysis he has thought of as a separate folder. And when the codes have stabilized and all that, push it up So he has complete control over whatever he wants to do. So I can actually see the changes on his repository on GitHub at a later time. Hmm? There's also a way to track on a separate branch. So you can create a tracking branch. GitHub is very sophisticated in these things. So you can create a tracking branch, which will allow me to actually look at what he has done on a separate branch. And if I like it, I can merge it with my branch. There are all sorts of things we can do. So in this course, I actually expect you guys, I want to see a folder. You can't just come and say, hey, I would do this and that, and I did all this coding in my local computer. It should be visible. That's at least what we want to get at. So that's a sort of a good understanding of what you exactly you have done. Okay. So now let me just go over this a little bit so that you guys have an idea of what is being done. So the README here is very helpful. It will tell you what is going on. So you guys should use README. If you look at this Power project codes, this is the one you should start with. Again, it has its own README. So what we do very early on is that whatever we require for just doing this power analysis, we save it locally within this folder. Okay, so these are all small files. Even though I have 120 trials and 300 seconds of data for each of the channels, the power spectral density or the time frequency plots, they are averaged across trials. So they are actually very short files. And for this project, we just need these short files. So there are some programs like run save data, which will save the data. They generally just create a local folder which keeps all the data that is required for this project. And the nice thing is that this folder is usually not that big. This is only 90 MB in this case. The data that's shared with you is about 40 odd gigabytes. So once you save this data, you actually no longer require the original data folder. Rest of the programs will just simply read and just display data from this. Okay, so actually I didn't have to share the complete the raw segmented data with any of you. You could have run all the programs in this folder because this saved data is also on GitHub. It's also shared with you. The reason I have shared is because I want to do some new analysis that we haven't done, for which you need access to the segmented data. That makes sense? And this is the architecture we will follow until this the segmented data is shared publicly. Till then, just keep it to yourself. At some point, we'll make it public. But until then, use it to just save the data that you want for your project and just only use that to go on. Okay, so now we will see. Let me see where I am. No, this is not where I want to be. I'll go to BK1. I'll go to programs. I'll go to Power project codes. And I will run, 1 second, I'll get rid of these things. Yeah, so run display power data all subjects is going to generate a GUI. Run, let's see if it works. And display power data all subjects. You are lucky. Yeah, at least it has run here without throwing any errors. Okay, so Let me just explain what is going on here. I can either compare the data in a paired fashion. So 30 meditators versus their 30 paired controls, or in an unpaired manner. So both, so there it's 36 meditators and 35 controls. I can choose. So let me choose EO1. So I want to look at the data for the EO1 condition. And so we assume that the 2.5 second data is divided into two parts, 1.25 seconds of spontaneous data and 1.25 seconds of stimulus data. In the EO1 condition, both are the same because there's no actual stimulus. So I can either choose this segment, that segment, or both the segments. So that's an option that is given here, or combined. So I'll use combined. And I can compare my power spectral density with respect to something. So I'll keep it as none. So then it will show you the raw power spectral densities. Here I can choose the frequency range of that I want to look at. So this is set to slow gamma and fast gamma, but I can change it to whatever I want. There are some cutoffs that we have. So in some, for every trial, let's say a person's eyes are open, the person does this task, we get 120 trials, but maybe the person was not looking at the screen, or maybe the eyes were closed. So there are lots of programs that we have already run to find out which of these epochs are actually useful. The remaining epochs are labeled as bad. So you may have seen this bad trials uh, date. So where is that? BK1 data, segmented data, bad trials. So this, again, there are other sort of various ways of, doing this pre-processing and saving this bad trials. It only tells you out of those 120 epochs that we have, which ones are worth using. And it's going to vary from not only subject to subject, it's also going to vary from protocol to protocol. So only if I have at least 30 trials, I'll proceed. Similarly, only if I have three good electrodes in the electrode groups that I have, I will proceed. Otherwise, I'll reject that subject. So I will not always have 30. I may have 28, 29, depending on which protocol I'm looking at. Anyway, so with luck, if I plot this, it should do something. Yeah. Okay. So this is the power spectral density for the occipital and frontotemporal electrodes. And in this case, we are just looking at alpha band, slow gamma and fast gamma separately. And for those three ranges, checking whether it is significant or not between meditators and controls. So these are the three power bands, 8 to 13, this and that. And this plot is exactly the one. Actually, let me just so I can change this frequency range to 30 to 80. Let me just do that and run this again. I can clear everything and plot. So these two plots, I'm recording everything. You don't have to take pictures. You can, and these two bar plots are exactly what's there in our paper. It's exactly this one. So you can actually recreate completely the plot that I had shown you here. Spontaneous gamma. This is EO1 condition. These are the two PhDs. And here, this and this is the change in power computed between 30 to 80 hertz, this range here, 30 to 80 hertz. So I can actually get these bar plots as well as, so basically whatever I showed you, you can simply see it here. Okay. The topo plots, this GUI, this is for the meditators. This is for the controls. And this is when you take a difference. So yeah, so this plot is that other topo plot that we are looking at. Okay, so once you play around, this might be a lot of information right now, but once you play around with it, you will get an idea of how this thing works. So between 30 to 80 hertz, this is the power in meditators, this is the power in controls. Control minus meditator is the plot shown here. So meditators have more power than controls in this broadband gamma range pretty much everywhere. Now, suppose I want to look at M1. So I can plot that and it will generate that M1 plot where again, meditators have more power than controls everywhere. Now, if you want to compare it with EO1, you can plot that also. EO1, if I plot, now you'll see that the two plots will be slightly below. Did not work. There. Okay, so this is the increase from EO1 to M1. Here in the I mean, here it's not dotted. In the figure that I showed, it was dotted, right? So these were the EO1s and the M1 is a little bit raised up. All of these things you can easily just do in this GUI itself. You can go to G2. Let's try G2. G2 stimulus. Now G2 stimulus, if you just plot, it's going to plot a raw power spectral density. This will not look that great. Again, there is a gamma here. It's just sort of not very obvious. What you have to do is you have to compare it with its own baseline. So reference, now you put G2's baseline so that it will plot the change in power between stimulus and baseline. Now when you plot this, you can see that meditators have more gamma than controls. In fact, this and this low gamma, this is exactly what's there in the paper also. And it's very localized to the occipital area. In the other area, nothing is happening. These are the corresponding topo plots. Okay, so this thing will allow you to generate all the plots that you want. So you just have to spend time. Might be a little overwhelming right now, but every plot in our that we are showing, you can easily generate by looking at this. M2 again, somebody was asking about M2. So going back. Yeah, so this is the one that I was telling you where the dotted ones were shown in EO1 and so this is that plot. This one. Let's try to get this plot. So spontaneous condition M2 minus G2 and stimulus induced M2. Let's see what these mean. So I'm going to look at M2. Within M2, I'm going to look at the spontaneous condition. Okay, and let me not put anything here. I'm simply asking during M2 spontaneous condition between controls and meditators, what is happening? So if I plot that, this is what it is. Meditators have more power than controls, something that we have seen earlier. But the key thing now to note is how is the meditation related power different from simply when their eyes are open, but they are not meditating. So I have to compare it with something, right? So let me compare it with, I could have taken EO2 condition, but in this case, I'm taking the G2 condition. It will remain the same. Nothing will change if I use EO2 also. But in the paper, I did the G2 one. So essentially, this is M2 spontaneous minus G2 spontaneous. This is the epoch where the eyes are open, but there is no stimulus on the screen. So when I compare this, then you see these topo plots. This is what I had shown. This is for the 30 to 80 hertz range, where meditators have this lots of gamma here, controls not so much. Okay, so this is the frontal gamma, and it's very prominent in meditators versus controls in the frontotemporal electrode. These are motion sensitive, so if you don't move, they actually don't do anything. When it comes near you. True. That's where the hypothesis is tested. Whether, but generally, if you don't move fast, they don't do anything. Yeah, easy to say. So this is that M2 minus G2. Then during epochs when there is a stimulus, so I'll choose the stimulus one here. And in this case, the reference will be M2 baseline. When I plot this, now I'm looking at the change in power in M2 compared to M2 baseline. And then you see this increase here, in the slow gamma range, which is much stronger than. Okay, so all the plots in the paper are right here. This one GUI, keep changing and you start getting this. Which one? So these three correspond to the frequency ranges. In fact, you can also, if you wanted to look at other electrode groups, if you change those, it will, let me see if this can be done. This program can do a lot of fancy things. But to answer your question before I get to that, this fourth one, this one simply tells you which electrodes are usable. I think that's what this one is. I forgot what this one does. This panel generally tells you how many electrodes are usable and all that. So you don't have to pay attention to this one. These ones correspond to the frequency range of interest. So this is the alpha suppression, meditators and controls, and this is meditator minus one. Okay, different. So if I was just going to see, so suppose you wanted to look at other electrode pairs also. It's actually, from what I remember, very easy to do. These are the frequency ranges. You can add more and the number of panels will change accordingly. Sort of cutoff, median, electrode groups. Yeah. Get electrode groups. By the way, if you are new to MATLAB and totally getting overwhelmed by looking at all of this, nobody learns MATLAB by reading anything. You learn MATLAB by doing things. And all you have to do, let's say you're just starting out with this. Oops, sorry. What have I done here? So suppose you start out and you simply want to know what this command does. So you can put a breakpoint. You can just click here, and when you run this, it will just come here. You can see what all variables are there. These are the variables. These are the sizes. And if you just want to run this, there are various ways to do it. So I can put a breakpoint here and let it move one step. Or I can just copy this and Control C, Control V here. So you will know exactly what that one line of code has done. So in this case, it would make this panel here. Sorry, I should have kept it here. Let me just do it that way. Now, suppose you want to know what this panel is, these three lines of code is doing. And then you do a Control C and a Control V, and look, it has made this one thing here. Okay, so it's actually very easy, and you just have to know how to use breakpoints and how to use help. So if you want to now know what is UI control, you just write help UI control, and it will tell you exactly what this command does and how to use it and so on. So the more you practice, the more you get. It's very easy that way because all you have to do is just play around with it. Getting back to this thing here, I'm showing only, let me see, where was I? Cutoff ranges, electrode group. So there's a function called get electrode groups, which can give me for the power one. Yeah, so I'm using occipital and frontotemporal. I'm telling how to create these, but you can also use other electrode groups. Okay, so if you want to look at what has happened at central electrodes, other electrodes, you just have to modify this program. In fact, by default, get electrode position on grid should give you um, five different electrode groups. Again, it depends on what kind of cap you have and what are their numbers. Yeah, so by default, it gives all five. So if you wanted to see some other area that we are not looking in this program, you're welcome to do all of that. It's very easy to change. Anyway, any questions about this? Yes. It's a heat map. And baseline, and then compare another with respect to that. The differences are plotted, the magnitudes that are there from -2 to 2. What exactly are the like change in power decibels? Yeah. So we convert all power to decibels on a log scale. So we are showing log of power. So when we subtract, it becomes a change in power. Log A minus log B is log A divided by B, right? So it converts it to decibels, which is a change in power on a log scale. So a simple subtraction, once you do a log, can be interpreted as a change in power. Okay, so which is what you are seeing here. So this is all in decibels, dB. Okay. All right. So see this, no substitute to this. You have to spend time playing with this code. Otherwise, things will not go anywhere. The good thing is all you have to do is just play with this code, put breakpoints, see how it's working. And then if you do it for a few hours, I think you'll have enough understanding to move forward. So let me just tell you a little bit about one more project. This is done by Vinod, and this is on connectivity. Several reasons. One is that Vinod is actually a TA for this course. So, and he's used this architecture very well. So he had this connectivity project folder where he did, so the codes there are written to do functional connectivity analysis, and it is done using field trip. So all the data is actually formatted to be compatible with field trip, which actually has a lot of other options that we have never explored. So you guys are welcome to try out some of those things like causality analysis, network topology, blah, blah. So lots of things you guys can do. This is an easy project because essentially you have the whole architecture with you. You are just changing some commands that will implement a different code. So functional connectivity, we will, we'll spend several classes on this later on. Let me just for now say it's a measure of how consistent are the phases recorded from two electrodes. Okay. So in suppose I have a seed electrode here shown in green, another electrode in yellow. I am simply asking that on a given trial, what is the phase difference between the two? And that would be a vector in this 0 to 2 pi this space. And across these 120 trials, if the phase relationship is always the same, then all the vectors will point in the same direction. Then when I average it, it will still be a big vector because they're all pointing in the same direction. On the other hand, if the phase are all over the place, like for this case, then the vectors will all be pointing in different directions. Then when I take an average, I'll get a small value. So this magnitude of the average vector is a measure of how consistent is the phase relationship between two electrodes. Okay, so at 10 hertz, it is very consistent, large vector. At 80, it's less. If you do it for all different frequencies, you'll get a plot like this. It's a measure of functional connectivity between these two electrodes. Okay, so we take these occipital electrodes as seed electrodes, and we actually, let's go back to this old data set with lots of subjects and see how functional connectivity changes in the brain as you are getting older. So what we have done here is that we have taken 6 different electrode groups that are separated by different distances from the seed electrode. Let me just go back here. These are the seed electrodes. These blue ones would be very close by. The green ones and orange one would be seed electrodes that are very far from the seed electrodes. So naturally, as you move farther away, you would expect the functional connectivity to be less. That is what is shown here, that as your electrode group moves away, functional connectivity falls. You also see that there is a nice bump in the alpha and this is gamma range. Question is, if you do the same for elderly people, what you will find? What will you find? So more connectivity or less connectivity as you grow old? Less. Less. Okay, you grow old, brain is less connected. So you can see it's in the alpha band and the gamma band, there is a reduction, but other frequencies are unaffected. It's only a reduction here and here. These black and gray ticks essentially show significant bins that are significant. Gray is without, I think it's 0.05 and black is 0.01 significance level. So not that surprising. Surprisingly, we see this even during baseline where we didn't expect a bump in the gamma range, but it turns out this bump is always there, whether you have a stimulus or not. Now comes the question, what happens during meditation? That's the real question. So let's look at EC1. Now, what do you expect? Meditators, more connectivity or less connectivity? More connectivity, right? Because we saw that it's reversing, blah, blah. So turns out there is no difference in connectivity. This is the connectivity plot. This is the power plot that I had shown earlier. So the power shows this expected change. Connectivity is unexpected. It doesn't depend. It's not different between meditators and controls. Boring result. But they are not meditating here. They are just keeping their eyes closed. Eyes open in this case. What happens when they meditate? Now, what do you expect? More or less when they are meditating? Answers are more, right? This is a very surprising result. Connectivity drastically falls between meditators. You see, this whole thing has actually shifted down. It's as if your brain essentially becomes disconnected. If you look at these meditators, this is the connectivity plot. This is for the controls. If you look at the distance, it's almost as if you're making islands here. So these are the seed electrodes, and it's essentially getting less connected with the rest of the brain. The power increases, but connectivity falls. Very surprising. So if you just compare the connectivity for meditators, for controls, when they are meditating and when they are keeping their eyes open, no difference. In the controls, it doesn't matter. But in meditators, it's a very drastic fall. So the meditators actually sort of stop the waves. The way you talk about, there are mental waves in a lake, and the goal of meditation to actually make the water still. Essentially, this is something similar to that. Chitta vritti nirodham. What about the effect of like certain drugs? Very good. LSD. Those are the things that we want to look at some point. In fact, Anchal, she was a PhD student. He really wants to do that. I don't think we'll get to LSD, but we will have plans to get to. We have plans to get to at least different types of anesthesia and anesthesia to start with. So yeah, these are all excellent questions for us to ask. Interestingly, if you take the seed electrodes in the frontal area, you see that the alpha connectivity falls. Everything else remains the same. So the brain actually gets disconnected, but in different frequency ranges, if you change the seed electrodes. All of this you can easily run with the programs that are there in the connectivity project. Okay, so as I said, it's run in a very sort of structured way. We have a single repository in which different projects are different folders. If you go to connectivity project codes, you should be able to, let me see actually it works or not. Programs, connectivity project codes. Yeah, again, there are some programs that save the connectivity data, then remaining programs run it. Let me see if this works. Tell you what, tell you what, so we'll have Vinod give a tutorial next Friday. I'm not here. Let him show all of this. Okay. So I'll just finish off because I only have 5 minutes with what you guys can all do with this data set. So see, as I said, this data set is still largely unexplored. There are lots of analyses, lots of things that we can do. And I'd really encourage you guys to take this seriously and try to do new things that we haven't done. We'll be discovering things together. So this is a summary of what we have done so far. I told you that we have looked at functional connectivity, but also source localization, slope of the power spectral density, and fractal dimension in the EEG, this TLSA data set. And these are all done projects with papers that are already out. In the meditation data set, we have done these things already. And this is part of the paper that's on bioRxiv. Fractal dimension, actually, there was one person who was supposed to do it, but she doesn't want to do it now. So it's a free project. If somebody wants to do it, you can do that. What else can you do? In the monkey data, we have been doing this burst dynamics, traveling waves, cross-frequency coupling. These are done by these students and postdocs. So burst dynamics is something that Vignesh has followed up in the EEG meditation data set. It's actually finding that meditators have longer bursts. Their brains produce longer bursts. It's a very cool result, but it's very preliminary. Bhargav, who was doing traveling waves, is my sort of like favorite project in some ways, because this mental waves that people talk about, we want to see whether in EEG there are waves like that. So Bhargav has started this, but it's something that if some of you want to do this, can take this up. So also cross frequency coupling that nobody is looking at right now in the lab, so somebody can do that. What else can we do? Bunch of other things, causality analysis. These are all correlational measures. Functional connectivity is correlational. You just say whether they are correlated, whether there's a direction, if this one is causing versus that. That thing is done using causality analysis, Granger causality being one. We again haven't looked at it. These are something that you guys can do. In general, this meditation subject or consciousness, this is related with a lot of very sort of interesting ideas related to complexity. Okay, so uh, well, it can also be done in terms of the electrodes, but it is more relevant for the EEG data set. Yes. So not everything is sort of useful for all these signals. So you have to pick and choose what you want to do. These are very easy ones to get into. So there is this idea that there is a fairly prominent idea. It's called the integrated information theory. That the consciousness is a sort of direct correlate of what is called integrated information. It is very hard to compute, by the way. But this idea of using measures of complexity to understand consciousness, different states of consciousness, and relating it to meditation, those are very interesting questions. Again, we haven't looked at it. You guys are welcome to do that. Finally, there's our thinking is that meditation changes the physiology also. You'll not only learn to concentrate, you also learn how to breathe properly, and the entire physiology couples in very interesting ways to allow you to get into this other states. And we are measuring all these other parameters also. So coupling with physiology is another very interesting question. So Vinod, who's the TA, has started looking at some of this. Again, this is an interesting project for some of you to follow. Eventually, we want to look at how, these are experiments in which there is no task. When you are coming out of, like anesthesia, or if you take a drug, or if you, take, listen to music, which takes you to a different plane altogether. This is where it's just like a journey. You start from some state, you go somewhere else, and all you have are these signals that allow you to sort of track this journey. So maybe in this high dimensional space, there is uh, some other space that you can create where these, as you are developing in your meditation or other things, you can track these things out. These are again sort of advanced things for us to look at. But eventually, this is what we want to get into, where a person goes through a particular state change. Could be meditation, could be effect of, let's say, brain stimulation, could be coming out of anesthesia, could be, as he said, some psychedelic drug, whatnot. We just need to get to a state where, looking at these signals, we are able to quantify these things. Okay. It's again up to you guys how much you want to invest and how much you want to think about this. The data, codes, and everything is now already given to you. I'm hoping you guys will take this up seriously, and we'll have some interesting projects by the end of this semester. Okay. All right, folks. So now it's all over to you. We'll start now with actual coursework from Monday onwards. See you guys.